another 20 or so people online as well. So this is the first in our 2022 series of the director seminars. And again, we're focusing, at least for the first half of the year, mostly on our own, try and share information about the research that goes on within RSB across divisions. So it's terrific to see you all. There are more chairs up the front here on both sides. So feel free to come up. Okay, so we've been going around different divisions, and today it's a pleasure to have Caitlin Byrt from Plant Sciences. So I won't go on with a long introduction. Kate's had a wonderful and varied history, moving between the University of Adelaide and Newcastle and back to Adelaide in her postdoc years. She did a DECRA, then that she was successful in a future fellowship, which she's on at the moment, which she took up here in 2019. Along the way, she scooped up numerous awards, including a tall poppy from South Australia. It's a good thing we don't cut them down anymore. The Peter Goldacre Award from the Australian Society of Plant Sciences. And she's just been promoted to level D in RSB, which is an achievement. And she's also the Deputy Director of Research in the ARC Training Centre, which of course. So she's very busy, and I think, as you'll see today, the work exemplifies moving back and forth between very fundamental cell biology, fundamental molecular physiology of plants, to applications, some of which you would never have imagined from a plant science. And I'll let her to fill in that story. And with her team now, she's starting up their own company to try and commercialize some of their discoveries. So that whole spectrum from fundamental science, translational, through to actually trying to start a company to further extend their work. So it's really exciting stuff. Caitlin.
Awesome. Thank you, Craig. And thank you everyone for coming. It's awesome to be able to see you in person instead of talking to a screen like we've had to do for the last couple of years or so. So hands up if you recognize this image. Yeah. So what's it an image of? That's right, the 14th of January, Hunga Tonga volcano. So NASA estimated that the explosive force of this was like the energy of 100 nuclear bombs going off. So to help people conceptualize the power of that kind of explosion, the ABC News shared this footage of, see if I can get to play, molten salt being dropped into water. So you can sort of visualize the release of that energy. Obviously, for a volcanic eruption, there's like all this pressurized lava, but they think that they're part of the power of the explosion related to the chemistry involved. So it's a cool visual representation of energy release when molten table salt hits water. So our lab team also studies salt and water and energy, but at a very different scale and a very different context. So our work is at a molecular level and in the context of living cells. So we study the function of molecular mechanisms involved in salt and water transport across cell membranes. And we look at how this relates to cell energy. So in this particular context, we often think about energy in terms of the voltage across the membrane. And we try and understand how these types of salt and water transport mechanisms are regulated in plants in the context of plant adaptation to really challenging environments like salinity stress and drought stress, and how we can design and engineer strategies to improve crop productivity by manipulating the function of these type of transporters. And we also look at whether or not we can learn from what plants do and borrow parts they've developed to be able to engineer precision separation technologies using modified versions of plant mechanisms. So there's a huge number of communities and plant scientists that support us in the context of creating collaborative opportunities, mentoring, supporting our work. But what I want to do today in the interest of time is focus in on the team in the lab at the moment. For many ECRs, the last couple of years have been super tough in terms of networking and being able to promote your science when you're trapped at home a lot and international conferences are off. So I want to promote the projects that our team were involved in. So we've got Sam, Anna Maria, Isabel, Shaggy, Lorraine, Preet, Taryn, Rose, Riley and Amy in the lab at the moment doing a multitude of different projects. And I also want to acknowledge the team at the University of Adelaide, Sharon, Stephen, White, whose work is also going to be represented in this talk, and thank the funding institutions that are supporting our various projects. But the common theme of these different projects, even though that many of them are on different types of transporters or different crops or with different applications, the common theme is that we build and test membrane transport mechanisms to contribute to the diverse toolkit that we anticipate needing in the future to be able to prepare ourselves for having food and water security. So I picked out 3 broad research questions that our little sub-projects feed into. So these are, how can plants best regulate iron and water transport mechanisms to adapt to saline or water limited conditions? How can a cell or an organelle adjust its iron or water flux to adjust to challenging conditions it experiences or to support the transport of metabolites or carbonates? And can the mechanisms that plants use to manage salt, nutrient, mineral, water flux be incorporated into separation technologies for harvesting resources from waste? So in the context of this first question, how can plants regulate iron and water transport to adapt to saline and water limited conditions? Why might we be interested in that? Well, by 2050, it's forecast that Australian saline land area may triple. In many places where the soil is saline, we lose up to 50% of the yield potential of our crops. There's ways to help crops to maintain productivity that involve influencing how plants, salt influx into plants, root to shoot transport of sodium and compartmentalization of salt ions in plants. There's also a multitude of other different ways to approach it, but for our work, we're going to focus in on approaches that relate to that. And I would like to acknowledge the influence of Rana Munns, Mark Tester and Steve Timon on getting into this type of work. So it's been about 18 years or so working on this topic, and their interest in it was absolutely contagious, infectious, back in 2005 or so. And so this is still like a major topic that I'm interested in working on.
Okay, sorry to interrupt. I just got a message from someone online. They said I can see your notes from this.
Ah, okay. How do I fix that? It must be screen sharing. Thank you, whoever online spotted that. Must be that one. Is that better online, folks? Sorry.
Yeah,
That's better. Brilliant. Thank you. Awesome. All right. Cool. So, so lots of the work in this area relates to figuring out how to control where sodium goes in plants. Thanks. Sorry. And so the outcome of this body of work so far is that we've been able to identify the molecular basis of some key mechanisms that control sodium transport. So here from like root to shoot in crop plants, and also the molecular basis of mechanisms that function a bit like a non-selective cation channel. So they let monovalent ions like sodium and potassium across cell membranes. So in both cases, these are mechanisms that were detected in terms of physiology, so the conductance was detected, but in both cases, the actual molecular identity remained a mystery for a long time. So it was very exciting to be able to deliver the molecular identity of some of these sort of important mechanisms that influence sodium transport in crops. So in the context of the root to shoot transport, a lot of this work has been on a type of transporter called a HKT, which stands for high affinity potassium transporter, which is confusing because subsets of them are functioning sort of as sodium transporters rather than potassium transporters. And then in the context of identifying candidates for non-selective cation channels, this related to identifying subsets of water channels called aquaporins that function as ion channels in plant cell membranes. Oh, the mouse has disappeared. There it is. No, it's all right. I got it. Awesome. So, and as part of these bodies of work, we were able to generate germplasm collections where we modified the function of these mechanisms towards understanding how plants use these mechanisms to adjust to dry and saline soils. And one of the most satisfying parts of this work has been that it has delivered real world impact. It's created options for plant breeders and farmers to select access to germplasm that actually can perform better in saline soils. So, for example, the manipulation of the HKT type sodium transporters that help plants to be able to limit the accumulation of salt in photosynthetic tissues where it is damaging and limits productivity, has actually been used across the globe to improve the salinity tolerance of crops like wheat. It's been used in tomato, in barley. Teams have been using it in rice as well. So limiting sodium accumulation is a useful strategy for introducing into crops that are growing in saline conditions, particularly where water is not limited. But we suspect that there might actually be a catch to use of this mechanism in conditions that are dry. So plants need to be able to ensure that they have sufficient internal solutes to be able to counter the osmolarity of the soil solution. So otherwise you end up in a situation where if it's very saline outside, the water gets sucked out of the plant. And so what this plot represents is some options for plants in the context of managing their shoot osmolarity over time in saline conditions, so in 150 millimolar. So if they exclude almost all of the salt from their leaf tissues, then to be able to osmotically adjust, they're going to have to use organic solutes. They're going to have to expend their carbon on creating solutes so that they can osmotically adjust. If they let a lot of the salt in, then they've got that solute available there, but it'll lead to oxidative damage, dehydration, and disrupt metabolism. So letting salt in can be a good thing because you can actually use it as kind of like a cheap osmolyte as part of your mechanism to osmotically adjust. Cheap, but not free. So there's a cost associated with it. So if you're letting passive sodium influx in, then in the context of being able to maintain the electrochemical difference across the plasma membrane, then you're probably going to have to spend ATP to pump protons out. So it's useful to be able to say, let sodium up initially so that it helps you osmotically adjust, and then put on the brakes once you've got sufficient to be able to manipulate what happens with that sodium to osmotically adjust, and so that you're not actually having to use up all your carbon resources to osmotically adjust. So one of the other key factors in the context of how you manage this is compartmentalization of the sodium, because if it's floating around free in the cytoplasm, it's going to cause damage with metabolism. So being able to stash it somewhere safe but still use it to osmotically adjust is important. And in the context of the relationship with water availability, we're trying to understand why it is that in saline wet conditions, it's very effective to use an exclusion mechanism, but it becomes problematic once you've got saline dry conditions. So maybe still being able to have that water flow enables you to kind of flush through and maintain hydration. So those are the sort of relationships that we're trying to understand to be able to design mechanisms for particular varying conditions in the environment. So we're investigating this potential catch to sodium exclusion in dry conditions. But one of the pieces of good news is that if water's not limited, whether or not you use a HKT mechanism and it's a non-saline condition or a saline condition, there's no yield penalty in non-saline conditions. So it is an inherently energy efficient mechanism to use for sodium exclusion. And in the context of energy efficient mechanisms for managing sodium, these are other cohort of candidates that we're interested in, as I mentioned before, are these ion channel aquaporins, which, so aquaporins form as tetramers in cell membranes. So they've got 4 monomeric pores and then a central pore, which is, we think, the pathway by which can function as a channel for monovalent ions. So we got really inspired by work of colleagues in the animal sector because sort of going back in time, a Nobel Prize in 2003 went to Peter Agre for sort of discovering aquaporins. They're found in every kingdom of life. There's 13 in your bodies, 2 or 3 in single-celled organisms, hundreds of different types in plants. And Agre's work indicated that some aquaporins can function as water channels that are highly selective for water and exclude all ions. So there's even been reports of aquaporins that can sort of transport 4 billion water molecules per second and exclude everything else. But there was a period of controversy when our colleague at Uni of Adelaide, Professor Andrea Yule, observed the human aquaporin 1 to function as an ion channel. So how can it both exclude ions and also function as an ion channel? So Andrea worked out that there's an interaction with cyclic GMP and differential phosphorylation of some of the key loops that can enable it to switch, so shut off the ion channel function or turn it on. So without that GMP interaction, you're not going to see it function as an ion channel. So this helped to resolve some of the controversy in the field as to why you would observe something to function exclusively as a water channel and then be able to also see it function as an ion channel. So Yule's team have linked the human aquaporin 1 function to things like cell migration and also to, it's a drug target because their team discovered that cancer cells use both the ion and the water channel function of the human aquaporin 1 to metastasize. So last week they published this data, which was very exciting for us all to see. It's single channel patch clamp data for the human aquaporin 1 in proteoliposomes. So you know, what you're looking at here is the control liposomes without the human aquaporin 1 and the liposomes with it showing the ion channel activity, which is awesome because there's always this fear in the context of studying ion channel aquaporins that when you observe conductance that it actually just relates to the aquaporin interacting with some other channel and somehow indirectly causing the ionic conductance. So it's like for all of our teams working on it, we're like, oh, how do we know for sure that it's actually this mechanism? So we're very excited, and this is something that we'd like to do as well. This is just a little indication of how you can take a little patch of membrane so you can just get a single channel and then be able to detect whether it's associated with ionic conductance. So plants also have ion channel aquaporins. So we observe there's at least 2 plasma membrane intrinsic type proteins or PIP2 type proteins in the model plant of Arabidopsis that are capable of functioning as ion channel aquaporins. So there's some conductance data there for those 2, PIP2 and PIP2 relative to controls. So this is detected by injecting oocytes with RNA coding for those proteins, and the oocytes express it, and then we use 2 electrode voltage clamp technique to measure the ionic conductance. So that's where you can clamp the membrane potential at different voltages and then measure the currents across the membrane, control and aquaporin expressing oocytes. So since then, we've been working on trying to understand what the physiological role is and how they're regulated, particularly in relation to plant adaptation to osmotic stresses. And this has been really hard because aquaporins do heaps of different things and respond to heaps of different things. So it's hard to actually tease out what's going on. So they're influenced by, changes in pH and calcium can affect the gating. They're influenced by ROS signaling, so H2O2, cyclonucleotides, hormones. Their localization can change in response to these sorts of things. They influence the transcriptional level, translation, post-translational modifications like differential phosphorylation and the stoichiometry, so the interaction between different types. So all these different things are going on in plants at the same time, and we're trying to tease out how on earth you would use an ion channel aquaporin, say, as opposed to an aquaporin that exclusively transports water if you're going to try and adapt to osmotic stresses. So one of the features that we're interested in is whether ion channel aquaporins are part of a process that can help move sodium inside small vesicular compartments. So it's been observed previously for PIC2-1 that salt stress causes differential phosphorylation in C-term tail, and that's associated with localization change of lots of PIC2-1 into small vesicular compartments. It's also been observed that a lot of salt goes into these compartments and that they interact with the vacuole, which I'm not really sure is showing up there, but it's here. So is this part of some way that you can actually keep the salt out of the cytoplasm, but somehow encapsulate it and actually shuffle it around as part of your method to osmotically adjust? So this is something that we're interested in the context of understanding how, there's plenty of examples of plants that can grow in seawater. So how are they doing it? And are these sort of strategies to be able to manage where the sodium goes important for those sort of mechanisms? So this work, which is not from our team, gives a bit of a context to some of the other complexities. So what's been shown is that those C-term tails on the Arabidopsis PIC2-1 ion channel aquaporin are differentially phosphorylated on a diurnal basis as well, and that's linked to hydraulic conductance throughout the Arabidopsis. So what you can see here is a light, dark, dark, dark cycle and wild type, as well as a 35S expression line of the PIC2-1, and they looked using antibodies to detect the abundance of PIC2-1 in different phosphorylated states. So we've got an osmotic stress changing the phosphorylation, we've got diurnal changes influencing the phosphorylation, and we've got the phosphorylation influencing the localization. There's also data showing that you end up with differences in stomatal aperture, because PIC2-1 is highly expressed in stomata, depending on the abundance of different phosphorylated states. So in these studies, I've used mimic and deficient versions of the PIC2-1 to have a look at some of the influences on different physiological processes. So to make things more complex, we observed that those same sites on the C terminal tail, which is the serine 280 and a serine 283, differential phosphorylation of those sites is likely to influence whether or not that you're in a water channel mode or an ion channel mode. So what you're looking at here is where we expressed phosphomimic versions and phosphodeficient versions, where we made mutations to switch the serine to alanine or aspartic acid and express them in the oocyte system, and had a look at the conductance as well as the osmotic permeability. And by switching what happens in the context of this C-term tail, you can actually push them to function like more as a water channel and less as an ion channel, or more as an ion channel and less as a water channel. So there's a bit of a relationship there in the context of those sites and what happens. So as I said before, we're dreaming about putting these in liposomes to actually know for sure what's going on and eliminate the potential for endogenous mechanisms to be influencing what we're looking at. So similar to what the human story, cyclic nucleotides also seem to be important, and lots of this work Sam completed during her PhD at the University of Adelaide. So she looked at the influence of adding kinase inhibitors like H7 to the oocytes, or adding treatments with cyclic nucleotides like CAMP and CGMP. There is a little bit of an influence just in control oocytes, so these are water injected oocytes for in CGMP treatments. But what was observed was that CAMP tends to result in an increase in ionic conductance. You get a decrease for H7 and also somewhat with CGMP based on the test rather than the ANOVA. So we've still got a lot of work to do to explore this, but what it doesn't do is tell us whether, like the human case, there's a direct interaction of the cyclic nucleotides, or whether the cyclic nucleotides are influencing endogenous kinase activity, because there are some conserved mechanisms through different kingdoms of life with cyclic nucleotide influence on protein kinase activity, or both. It could be both. So this is some of the things that we want to be able to understand, particularly because in lots of previous studies, there's links between what happens with cyclic nucleotide signaling and plant responses to osmotic stress. So it's known that cyclic nucleotides like CGMP inhibit the activation of non-selective cation channel mechanisms, and hypothetically, this could be also something that's been observed with CGMP in the context of the ion channel aquaporin regulation. So moving on from Arabidopsis, this is also a feature in cereals. So I'm just going to give one example because it's emerging that lots of different plants have ion channel aquaporins. But for barley, we worked with a team over in Japan to look through the barley PIP2 type proteins and observed that a barley PIP28 functions as an ion channel aquaporin and is also influenced by C-terminal tail phosphorylation in the context of the regulation, like a switch between whether you're going to function as a water channel or an ion channel. So leaving behind some of that whole plant stuff and zooming in a little bit, how can a cell or an organelle optimally adjust ion or water flux to adjust to challenging conditions or to support transport of metabolites and carbonates? So in this topic, we're actually, we're not being prejudiced about systems. We just want to understand what's going on. So yes, we're looking at roots in the context of ion channel aquaporin function to potentially let potassium in to access nutrients, let sodium in to osmotically adjust. We're also interested in the roles in guard cells, like whether it's some of the signaling processes that are influencing guard cell opening and closing. But we want to understand it from a cell biology type perspective. And so we're looking into things like yeast and connecting with teams within RSB who study Plasmodium. So, in Arabidopsis, there's 27,000 proteins to deal with. I think there's only about 500 in Plasmodium, and a lot of work's being done to narrow down which ones function in potassium transport. Yeah, just correct me if I'm wrong, I'm guessing. And so if you've got this sort of cohort of understanding of what's influencing ion channels function in there, and you can manipulate what happens in the context of potentially knockout ion channel aquaporins, that's part of a way that we can sort of more closely understand what's going on with their function in terms of cell expansion, swelling, the role of different kinases and turning them on and off through phosphorylation, whether or not they're helping in the context of adjustments to changes in osmolality, how they use the water and ion channel function in the context of adjusting to stress, whether they're part of an emergency pressure valve release. We're also interested in the context of features like their roles in nematode resistance, their roles in mangroves in the context of salt excretion from leaves. And in yeast, there's similar to the plant situation, there's been a long period of time of people detecting physiologically a non-selective channel activity that had no molecular identity. And our hypothesis is that the yeast aquaporins could be contributing to that because we have data indicating that that might be the case. So we love learning from discoveries of aquaporin regulation from other systems. And so this is one from Aston Uni, Rosalind Bill's team, where they look at the human aquaporin 4 function. So it's critical in the context of brain swelling. And this is a model that they generated in the context of the regulation of aquaporin 4 function, showing the influence of calcium signaling, cyclic AMP signaling, protein kinase activity, influence on aquaporin 4 phosphorylation and localization. So their team demonstrated that the human aquaporin 4 has critical roles in brain swelling that have important implications for medical approaches to managing brain trauma, which affects millions of people every year. So severe injury to the head can lead to the accumulation of water and swelling, which can be fatal. And what they've found is that they can actually use pharmacological treatments that influence the aquaporin 4 localization as a way to help patients to minimize that swelling, to buy them more time rather than drilling a hole and letting the fluid come out, which has dangers in itself. So some of these types of regulatory features are conserved across different kingdoms of life. And so we're interested in the context of the plant side of it and things like plant cell swelling, what might be going on. So this is a bit of a hypothetical model representing some of the gaps that we're trying to fill in the context of plants, of understanding how different signaling influences the permeability to water and ions, and how that switching of turning on and off is influenced by cyclic nucleotides and protein kinase activity, and how things come together in the context of the permeability and the localization to result in important physiological features. So some of those are things like, as I mentioned before, guard cell function. It's already been reported that, say, for example, the PIP2-1 ion channel function is critical in the context of lateral root emergence. And then we've also been working on looking at what happens during germination. So we are talking about a protein that's very abundant in cell membranes all across plants. So at the top here, looking at not our work, but work from a student who hooked up the ATPIP2-1 promoter to a GUS. So that's what you can see in blue. So that you can see how abundant it is in Arabidopsis. And then here, GFP is used in each of these examples so that you can visualize where the PIP2-1 is and its abundance. So say, for example, in the lateral root emergence example, if you overexpress PIP2-1 or knock it out, both of those impact the lateral root emergence, either in the context of delaying it or slowing it. So somehow that coordination of what the PIP's doing for solute flux is key in getting optimal lateral root emergence. So some of our work involves trying to understand in different contexts how different kinases come in and switch that water and ion channel function of PIP2-1 to be able to influence what's going on. So earlier I showed the examples of how those phosphorylation states of PIP2-1 are linked in the context of what happens to somatal aperture. And this is a different body of data from a different group, also showing that whether you have a knockout, so that's a PIP2-1, missing a PIP2-1 or overexpression lines, you end up with different things going on relative to the controls in the context of stomatal aperture. So we've been studying how different key kinases influence the function of the water and ion channel feature of PIP2-1. And then some of this work here is driven by a PhD student, Fan, who's in the University of Adelaide. And she was looking at what happened in the context of germination using material where we've either lost the function of PIP2-1, so knockout mutants, or have overexpressed PIP2-1 using the B5S promoter. And so with her work, we observed that whether you lose the function or overexpress it, you end up with longer time to reach 50% germination. And there are also implications in the context of the percentage of germination, less so in Hessus-Hartmann-Ness control conditions, but once you actually start to go into a situation where you've got saline conditions and those seeds are trying to germinate in saline conditions, then you end up with a reduced germination percentage, whether you've lost the function of PIP2-1 or overexpressed it. And then like this also interacts in the context of some previous work from this Footer group here, where they showed that another type of aquaporins in the tonoplast are critical in the context of Arabidopsis actually still being able to successfully germinate in osmotic stress conditions. So we really want to understand how different plants are actually using these kind of mechanisms in the context of being able to germinate in really, really tough situations. So stepping away from aquaporins, but still sticking in the context of ion channel activity, one of the things that's really exciting about the work that lots of our team do is that we're being given opportunities from colleagues here in RSD to look at what happens in the context of ion channel activity that relates to how plants can actually successfully manage optimal transport of things like metabolites and things like bicarbonate, and also how things like the use of sugar and ion transport type proteins can actually influence important features, so like rust resistance. So we're grateful to be able to collaborate with the C4 Rice Consortium and also feed into some of the work in the context of the bicarbonate transporters, as well as having a look at ion channel aquaporins in the context of symbiotic root modulation, and look at some transporters that switch between sugar and ion transport that are candidates for influencing leaf rust resistance. Because in each of those cases, again, we're looking at features where you're aiming for an absolute optimum situation of how you manage your resources and move them across membranes. So in the context of C4 rice, we're looking at what happens in C4 crops and then trying to re-engineer that into rice. And then also in the context of rice, trying to understand how this amazing system that algae uses in the context of being able to have the photosynthesis, how those mechanisms can be used in the context of building or engineering that type of mechanism into plants. So just coming into the last sort of section in the context of looking at whether mechanisms that plants use to manage salt and nutrient and mineral and water flux can be incorporated into separation technologies for harvesting resources from liquid wastes. Why are we interested in that? So we face a huge number of water and resource security challenges. There's already 2 billion people who lack safe drinking water. The World Bank has estimated that by 2030 there will not be enough water to meet demand in most major cities. In 2019, here in Australia, many towns were paying $1 million per month to just truck water in because they didn't have any. And even though along the East Coast we've got some rain, there's still 50 New South Wales schools who rely on bottled water because they don't have access to clean drinking water. We also have a crisis resource-wise in the context of nutrients. So in the last 12 months, the cost of like nitrogen, phosphorus, potassium nutrients have gone up from, in some cases, 100% to 300%. And lots of the nutrient resources are in demand, both for fertilizer, for food security, as well as in the context of renewable energy. So everyone wants urea for green hydrogen. So there's a lot of pressures on all of these systems. So historically, we've relied on ecosystem services to do a lot of our cleaning of our water. So 50% of global precipitation passes through plants every year. And plants are amazing, right? Like they're these solar powered systems that take in dirty water, create clean water, fix atmospheric carbon, produce oxygen, but we're overloading them in the context of the ecosystems in the context of the sort of waste that we create, and we create waste that they can't deal with. It's beyond the biology to be able to deal with it. And yet we face a situation where our clean energy systems that we need in the context of climate change depend on access to minerals and nutrient resources so that we can switch to electric cars and have, battery storage technologies and green hydrogen. So all of those place a huge demand on access to minerals and nutrients like lithium and phosphate and urea. And all of these resources are, they're out there, but they're in complex mixtures. So to get them requires separation processes. So our team formed a membrane transporter engineers to try and work on advancing separation technologies to be able to sort of support initiatives to have environmentally friendly ways that we can actually access and separate critical resources from like wastes and sewage, because in all of the cases of being able to access these things, it's only valuable once we actually get it into that pure form. And lots of the technologies used for harvesting these sort of resources are not environmentally friendly. So we got really inspired by technology development achieved by NASA and also in collaboration with a Denmark company called Aquaporin. So they looked at aquaporins in nature and some of their magic features, and they incorporated aquaporins that function exclusively as water channels in membranes inside filters so that astronauts can recycle around 93% of their bodily waste into clean water. So the way that tech works is that you input the dirty water and you'll be able to pull out clean water, but everything else in that dirty water remains in an undifferentiated form. And then this has been really successful, and Denmark companies spun it off into fancy under the sink units. This sort of technology is being scaled up, particularly in places like Singapore, where they want to use it for desal, so for their future water security. So we admired what they did, but we decided we want that system to be modularized so that you put in your complex waste solution, whether it's like mine tailing waste or sewage, you actually pull out key target resources in modular steps by controlling the separation of the layers in those steps, while still actually generating clean water as an output. So that's what we're trying to work on by engineering the selectivity. So this is our team, Sam and Maria up the front there, founding team working on developing this. And I also want to put a huge shout out to Lauren DeFall from the VEC team here at ANU, because she's been enormous support in helping us trying to get this initiative going. And this is just short.
At home, you can separate your household waste so that glass, cardboard, aluminium and plastic resources can be recycled. Is it also possible for organisations to recycle resources from their liquid wastes? Lots of industries like mining generate resource-rich liquid wastes. Demand for critical resources like lithium and urea is growing, but availability is limited. Producing these resources is inefficient in water and energy use and damages the environment. Our membrane transporter engineers team are developing novel separation technologies that can harvest valuable resources from liquid wastes. We engineer highly selective components for industrial membranes that control selectivity, allowing for modular separation processes so that membranes can function like a molecular sieve. Installing selective components that capture lost lithium, phosphorus, potassium and urea into membrane separation technologies is a key step towards sustainable resource management and transitioning to a circular green economy.
Last word's economy, kind of cuts off. Anyway, I guess coming back full circle, we've got no shortage of issues that cause water quality problems every time we have 10-year droughts, every time we have bushfires, every time we have floods, and you know, the poor people of Tonga, all their stuff's full of ash at the moment. So there's all these challenges in the context of being able to separate critical resources out of the enormous amount of waste. And so this is an exciting area to be part of. There's I guess a lot of groups who are trying to learn from nature to create bioinspired membranes where we use biological processes and re-engineer them so that we can access the materials we need for fertilizers and sustainable energy. And in the context of the membrane separation area, because of all these pressures and this demand, it's an area that's rapidly growing. So in the context of the market growth, it's growing by 9.2% per annum and is expected to be worth US $43 billion by 2027. And finally, last slide, I want to say that this is an awesome place to do this. So Canberra is an awesome place to do this. At the moment, there's like a huge amount of support. We're really grateful for the support from CEAT and Sebring teams and lots of mentors that have helped us along the way, but also to be able to tap into the extraordinary expertise in RSB in relation to studying membrane transporters, in relation to progressing synthetic biology and the superb tools that everyone is inventing and using. In the context of, like, as you can see, with engineering for separation technology, but also trying to progress the plant side, being able to tap into the expertise here in the context of photosynthesis and energy in plants and in the context of cell signaling and cell development. It's just an amazing place to be able to connect with different teams who are leading the world in these sort of areas, and teams that are also feeding into important areas of impact for ANU, so in the context of food security, soil, water, and environmental sustainability. So thank you all for coming, and our door is open. We really want help and support to try and keep things going, and also to be able to support other people's amazing initiatives as well in the context of some of the tools that we've been able to develop, whether they're applicable. So thank you. Happy to take questions.
There's plenty of time for questions. I'll take a couple from the floor, then we'll look online. So, that sort of has the breadth to it all. Yes, Stefan.
Maybe I have a question. When you have these artificial membranes for water separation, how do you stabilize the membrane is still suitable for biological pheromones?
That's an excellent question. So the systems.
Sorry, you might repeat the question.
Oh, yeah, sorry. Stefan's question was about how the membranes are actually stabilized in the context of how the kit works for the separation devices. So lots of the teams who pioneered this work, so the NASA teams and the Aquaporin teams and some other groups, because they don't have an exclusive thing on it, there's other groups that do it too. They create like nanotubules where there's like a polymer matrix that actually supports the membrane layer. So the membrane layer is attached to a supporting polymer structure inside nanotubules that wind around. So there's a lot of patents in that area and that's not an area that we want to develop. We only want to single in on engineering the parts, but it is an area that we need to understand. And it's sort of challenging because they kind of keep a lot as trade secrets.
Just
In the beginning, you're talking about the HKP one stuff. How salty or water are we talking about? Whether it's salt water trade-off.
I mean, that's an excellent question because I mean, there are cereals that cereal crops that can grow in like half strength seawater. So if seawater is around like 500 millimolar, like that can grow in 250 millimolar. So somehow they figured out how to do that. And of course, from an evolutionary perspective, you've got like, whatever types of algae that eventually crept onto land, got land plants, grasses climb a fair bit later, and then grasses climb back into the ocean because that's how we got sea grasses. So it's like biologically it's possible, but actually just narrowing down on what mechanisms are involved to achieve that is a little bit tougher. But there is seagrass genome now, and quite some differences in the context of aquaporins in it.
You're embedding aquaporin investments on embedding proteins into the membrane. How stable are the proteins themselves? Is that a concern or an issue? How reactive are those?
That's a great question. And something that you would feel intuitively concerned about it, like when you sit there and think about protein degradation. But in the context of when the NASA teams wanted to develop this, part of their end goal for what they wanted this device to do is to have minimal maintenance and last as long as possible. So the previous filters that they used in space let contaminants through that made the astronauts sick, got clogged up, and required so much maintenance that it cost a fortune always to get people up to the International Space Station and deal with it. So they figured that by using a system that works well in biology, so say, for example, our human kidneys and their aquaporins with, sort of quite a functional system in the context of managing water flux, that they would be able to build something that would actually last quite a long time. And it seems that they've been successful at that in the context of building something that requires minimal maintenance. So their post studies have shown that it lasts for a long period of time, not forever. But then you've also, I guess, faced that issue with industrial waste of other things in there that could cause degradation. So I think, again, like similar to what Stefan's question, having almost like a pre-separation before you get to the more specialized separation is important so that you can actually modify what is reaching your target proteins. But the group in Denmark have been able to do that and successfully create tech for managing things like, how textile waste industries, like the dyes and stuff, there's a whole pile of horrible toxic things. So they have spin-offs that enable people to install units to be able to manage those dyes that use the aquaporin system. So I think it's just a matter, again, of the modularizing so that you actually use whichever technology addresses a particular goal along the way to reach the end goal. And with that in mind, we've sort of been aiming to connect with some of the groups who develop other types of filters, because you might use something that's less selective but removes some things initially, but then move into the type of thing that we want to do, which is really fine control over the selectivity.
Michael?
I guess just a question about the saline salt plants. When you engineering it, does that then increase the salinity of the soil or is it neutral when you get to the soil? I just wonder if you have a feedback process where the saliva on it, where they make the soil more saline and you're creating new problems.
That's a great question. Oh yeah, sorry. So Michael's asking about you know, if you basically are excluding lots of the sodium in terms of the crop, does that just mean you end up with more in the soil? Whereas, you know, there's also initiatives where people are like, can we actually use plants for bioremediation as to try and like suck sodium out of the soils? So, I mean, in the context of like some of the key food security crops, if you're looking at, you know, things like wheat and rice and barley, you know, there will be just, I think, an upper limit to where it's feasible to actually use those crops in the context of the soil salinity, unless we can borrow some really magic mechanisms from some of the sort of super salt tolerant plants. But you will always face some sort of trade-off, is in that sodium has to go somewhere, right? Like, so you can optimize, but it's intricately related to what happens with water. So, you know, where we like admire some of the mangrove examples, as long as they continue to be able to access water, they seem to be able to manage to deal with it, like a flushing effect. There is some rainfall flushing effect in lots of these areas. So the salinity level varies a lot throughout a season. So it's going to be a matter of modeling the seasons, understanding the seasons, and working through the process of engineering the crops to be able to have the right mechanisms expressed at the right time, and implement other strategies. So, I mean, there's lots of things like, say, for example, adding pig manure to really saline soils does like a magic job of managing to limit the effect of the salt on the crop that you subsequently grow on it. So it's sort of like one little piece of big puzzle.
Can we see if we have any questions online on Zoom?
Yeah.
Or can anyone speak up if you're online?
It's not showing in chat.
Sorry, can you see the chat on Zoom?
Hiding. It just says settings.
Bear with us, folks. Well, which is why we fiddled with that. Any others from the, yes, Ben.
What's the natural range of obviously you have the water
we're desperately trying to work that out with working through a series of solutions. And one of the things that is interesting is that we wondered whether it's just going to be the same for all of them, right? Like in the context of sort of some of our initial work had shown that there's greater selectivity for potassium than sodium, which you might expect, right? Like plants want potassium, you know, they're not going to ever be short really of sodium. But that doesn't look like it's the case across different versions. So there is variability within the different ion channel aquaporin subsets from different plants in the context of their selectivity. And there's types of aquaporins that do unusual things like aluminium malate. So there's a number of substrates that the different aquaporin sets are permeable to grows like all the time, weekly. Within just seems to have been I guess a family where gene duplication and divergence has meant that you've actually been able to take this sort of function and then adapt it for a multitude of different purposes to achieve different things in plants. But we're definitely interested in like why not just use a potassium channel or a sodium channel? What's the advantage of having four water channels paired with it? You know, so our collaborator in Adelaide, Steve Timon, is like, you know, maybe it stirs, like has some sort of stirring effect of like you've got this great flux associated with the water movement. How does that interact with how you're managing ion permeability?
Up there, so just don't know how to get to this. You just click on the chat function. Yes, Q&A.
No, or Q&A.
Maybe stop shooting.
Now it's back here. Cool.
Repeat the question.
Okay, now I'm going to get through. From Joe Brock in the context of using computational design for engineering aquaporins, and we would absolutely love to. So that's a particular area that we want to reach out to other teams who are taking those sort of approaches to learn a bit more about what's feasible. Because at the moment, like I guess in the context of the strategy, we've got a series of amazing high throughput assays for testing substrate permeability that Sam and Anna Maria have developed and are able to use for taking huge numbers of aquaporins and teasing out which ones are permeable to what substrates, so that we can actually group them together and have a look at sequence structure, function relationships. And then also we have, I guess, the more targeted approach of like to electrode voltage clamping or the dream patch clamping to be able to look at individuals, which is much less high throughput, but then would definitely be keen to be able to sort of take that AI computational design type approach to see whether we can do things like have a guess at how we could engineer it for a particular function and then actually test in the lab whether our guess matches up with reality. That would be great. Yeah, please yell out if
So along similar lines, has anyone tried experimental evolution or directed evolution, say in a yeast system or whatever, to try and get to where you want to go?
So,
I thought about it, CO2, to sort of piggyback on the sensitivity system. But yeah.
So this is sort of the beginning, not the end, isn't it? There's so many possibilities.
yeah,
So
and
I-
so many unknowns.
Maya,
So excited. I'm looking at the aquaporins.com website. So cool. The one thing that I'm trying to get my head around is how in kidney you would have a nucleus and RNA and then constant replacement of used up aquaporins.
Yeah.
How does that work with those membranes? I mean, obviously, you guys just put protein by protein into a membrane. I imagine it's not some automation to that. And then what? They are used up to replace those. What's the how long?
That's a great point, Maya. So in the context of, I guess, a living cell system, there's a couple of things. So of all the different proteins, that aquaporins are some of the most stable, long-lasting proteins. So of the types of studies where they're like, how long does a protein hang around? Aquaporins are used for a long period of time before they, say, be degraded and recycled. So that's an advantage. But then, we're not talking about building something that has an infinite lifespan. So, say for example, in South Australia, where periodically our supply of Murray River water tastes like muddy river water. Lots of households would have this thing called a Puritap, where they have a little filter, which is a different filtration technologies, it's size exclusion sort of thing, but that has to be changed annually. If you can, as long as you can keep it more than like lasting for at least a year, if not more, then that's as good as it gets in terms of that kind of filtration.
Those products can work for a year, like the same protein keeps working for a year.
Yeah.
It's fine.
Yeah. That was one of those key factors, as we said before, for the minimum maintenance of the International Space Station. Yeah. It is pretty amazing. That fusion of biology.
so did your interest develop from growing up with Adelaide water?
Probably, yeah. I mean, one of the things.
If anyone's not tried Adelaide water.
Yeah. We also have a lot of problems with, so when they switch on the desalination systems are really poor at removing boron. So all our plant experiments tell us before anything else tells us that there's a problem, because we start seeing boron toxicity symptoms in our plant experiments, because it feeds through like the spikes in boron in the water is amazing and correlate perfectly with the use of the desal system, which is another area that we're interested in, because boron's an emerging key resource for battery technologies. So some of the studies in the context of the stability of battery technologies for reducing how likely they are to catch fire and explode, boron's becoming a key resource. So what, you know, we're interested in being able to multitask and basically be able to specialize. And of course, there are a bunch of boron transporting aquaporins that are specialized for that purpose. Thanks,
It's
nature.
Project 56, I think.
Yeah.
All right. So incredibly wide ranging. I will have to draw it to a close. I'm sorry those still have questions because we're up on time. Thank you so much, Caitlin.
Thank you for the opportunity, and thanks
Good luck
everyone for
with it.
coming.